Hi everyone, my name is Elias Quijano. I'm a Blavatnik Fellow with Yale Ventures. And prior to joining this team, I spent the last 13 years really thinking about non-viral drug and gene delivery platforms. And what I've come to appreciate over time is that while many of them can be deployed for systemic disease, few, if any, really penetrate the central nervous system. At B3 Therapeutics, we're developing a novel polymeric system that's specifically designed for intrathecal administration, exposing the central nervous system directly to our therapeutics that are encapsulated. Thus far, we've shown that these polymer nanoparticles can encapsulate small molecule drugs as well as oligonucleotides. And so far, our in vivo proof of concept data supports the development of this asset for treatment of brain tumors. Our company has been founded by academic entrepreneurs. Mark Saltzman's, work in drug and gene delivery has gone on to form the basis of Stratify and Xanadu Bio, two Yale spinouts. Likewise, Ranjit Bindra, physician scientist at Yale, has gone on to found Cybrexa, Alphina, and Modify Bio. And of course, Kevin Rakin, known to many in the Yale community, has been a significant investor and co-founder of many of these Yale spinouts. Raman Baha has recently joined the team as well, bringing to bear tremendous expertise in synthetic chemistry. And lastly, I was able to join this team having recently founded Geneo, which is advancing a monoclonal antibody for nucleic acid delivery, again, another Yale spinout. So when we think about brain tumors, it's obvious that there's a profound unmet need, be they diffuse pontine gliomas, medulloblastomas in children, glioblastoma in adults, or metastatic disease secondary to breast and lung cancer. What's common among all of these tumor types is that patients suffering from these conditions have poor prognosis as well as low survival. The primary barrier to effective therapy in these settings is the blood-brain barrier, which has been designed biologically to prevent the accumulation and passage of small molecule drugs. To overcome this BBB barrier, we and others have explored the idea of directly introducing drugs into the central nervous system. This has been done through convection-enhanced delivery. Here on the left, you can see an image where a radiolabeled tracer has been infused directly into the brain by CED. And what's obvious is that although the distribution is efficient within the first few hours, by 48 hours, the molecules have rapidly cleared. Likewise, intrathecal administration directly into the CSF has been explored. However, clearance of particles from this system happens on the order of hours. To overcome these limitations, we've gone ahead and advanced a polymeric nanoparticle platform, which not only rapidly distributes throughout the CNS following intrathecal administration, but has shown significant retention following IT administration. Here on the bottom, you can see fluorescently labeled nanoparticles that once administered IT rapidly distribute throughout the spinal cord and brain. And what's notable about these nanoparticles is that they're persistent. They actually last for up to 7 days following IT administration. When we take these spinal cords and section them, we can see that these nanoparticles rapidly associate with the meninges of the spinal cord and in many cases actually traverse into the central canal. On the right, you can see an example where we've now used PET imaging to trace the circulation of these nanoparticles once administered. And what you can see here in this model is that the particles rapidly accumulate within brain tumors. In this case, we're looking at a mouse model of medulloblastoma. Importantly, these particles are still detectable up to 21 days after a single IT dose. Using these polymeric nanoparticles, we've gone on to encapsulate DNA repair inhibitors. Specifically, we used for this example BMN 673, which is a PARP inhibitor. Here, using a mouse model of medulloblastoma. On the left, you can see we've introduced BMN 673 as a free drug, and you can see from the red line that no significant survival is appreciated beyond control. However, when this drug is encapsulated into our nanoparticles, you can see significant enhancement in survival as noted by the green survival line here up to day 100. We next sought to investigate whether or not this therapeutic approach was synergistic with temozolomide, the current standard of care. And what we found was that at day 100, this combination therapy actually resulted in 80% survival probability. While our lead indication and our lead product is really focused on developing polymeric nanoparticles to encapsulate and deliver DNA repair inhibitors, we have a number of exploratory programs that are ongoing, including the encapsulation and delivery of antisense oligonucleotides, DNA oligonucleotides, and plasmid DNA. as we continue to advance the technology and seek external support, we're really seeking to round out our set of in vivo proof of concept studies. And what we're primarily looking at doing this year is conducting non-human primate studies to really study the safety and translatability of this approach. Thank you.